Hello and welcome back to our science sip. It's been a while since we've sipped.
It's good to sip again with
It
you.
is good to sip again. So we're on a bit of a no alcohol kick for a while,
Yeah.
but that doesn't mean we're not going to have some fun. We made a mocktail.
Well, you made a mocktail.
I made a
which
mocktail.
I get the joy.
Yes,
of
it's
trying out.
strawberry ginger lemonade from Bon Appetit. And it tastes very summery because we're just hoping for that summer weather soon.
And that strong kick you get from the ginger does a nice job of replacing the alcohol.
Okay, if you say so. So what do you have to talk about today?
Well, I've been looking at the Journal of Lipid Research and the History of Cholesterol Medication
Oh, all right.
in America and in the world.
Okay.
It's been pretty exciting. I'm someone who deals with high cholesterol, and so I've been interested in looking at how cholesterol and the history of how cholesterol was tied to atherosclerosis.
All right.
From the early 1900s, people who had diets high in meat, eggs, and milk were generally associated with having high cholesterol, but it wasn't a one-to-one correspondence. Not everyone who had that kind of diet actually had symptoms that could result in coronary artery disease.
It's because of this pesky thing called genetics.
Exactly.
That means we're not all exactly the same.
Exactly. And I think what doctors are trying to do is come up with a way to be able to say, if I can look at your blood and take an indicator in your blood, like a blood test, and correlate that with your potential increased risk for coronary artery disease.
It's called biomarkers.
Yes.
Looking for a biomarker to be able to predict somebody who's at higher risk for heart disease.
And it's pretty exciting because you can appreciate this a lot more. I'm an electrical engineer. I don't understand the fact that lipids and complexes with proteins are very complex. Like the behaviors of these proteins cannot be,
Lipoproteins.
these lipoproteins cannot be predicted very easily. They can change in their density while you're measuring them. And that was something that was happening with all of the different categories of lipoproteins that presented themselves as a total cholesterol picture of your blood. So, one example of that is that the doctors would try to separate the fat from the plasma and see if they could use that to measure cholesterol. There was also tests where they tried to feed various laboratory animals extremely high cholesterol diets, only to find out there's cholesterol resistance in dogs.
Yeah, this is the other problem with research is that humans are not dogs or mice or hamsters or whatever. I mean, laboratory animal testing has, its role, but it's really hard to generalize the results to humans most of the time.
Indeed, especially when it comes to diet.
Yes.
We are so different from the animal kingdom when it comes to how diet interacts with our health. And one of the things that was discovered through the invention of the ultracentrifuge was a way to finally create that stratification of the different proteins in people's blood. And what they just started to discover was that there was not just one lipoprotein or a couple of lipoproteins, but that they could be segmented into 4 or 5 different categories. And what they decided to call these were high, intermediate, low, and very low density lipoproteins.
Right, because they're separated by centrifugation, which separates things by density or weight.
Exactly. And those lipoproteins now, once they were identified, those individual categories of lipoproteins could then be correlated, which one could then correlate to your chances of having a heart attack. So I found that to be very interesting. And I also noted that even though that research moved on from there and unfortunately initially was rejected by a lot of people because of the complex behavior of lipoproteins in liquid or in plasma, that it eventually, even the authors of that original seminal paper eventually had to fight to get that published in the Journal of Biochemistry. In 1985, the Nobel Prize went to additional research being done by two people by the name of Goldstein and Brown,
Mm-hmm.
who took that low density lipoprotein research, LDL,
Mm-hmm.
and discovered receptors. And through that work, the idea of developing statins eventually came around, which is how we treat cholesterol today, among other ways.
Among other ways, yes. Well, that's very interesting. I have something a little bit different, as usual.
I know.
Did you know, Dima, that about 38 million people in the world live with HIV?
Wow.
Dima spit out his drink. Yeah, so it's still a huge
big
problem. Yeah, I mean, I don't know about you, but growing up when we did, I think we both remember the 80s and 90s when there was a lot of coverage of HIV and AIDS, and
It was in the news.
in the news a lot. There's a lot of activism. Some of that has receded.
Yeah.
Some
We
of that
don't
has
see it
receded
much.
from sort of the public sphere, but HIV research, treatment, advocacy are all still going strong because the disease is incurable.
Yeah.
Except it's not exactly incurable. It's just the way that you cure it is really challenging.
And there have been a handful of examples.
A handful of examples, and there's one more now. So
Oh, wow.
a woman has been HIV free since 2017, and you're having challenges with the cocktail. Oh, maybe you should just sip it gently.
I love the strawberries.
I
Okay.
know.
Sorry about that.
You're being distracted. So there's a woman who's been HIV free since 2017. So that's not just low HIV virus, but completely undetectable. Yeah. And she had a transplant of HIV resistant stem cells. And the reason that she was able to do this is because she also had leukemia, and blood stem cell transplants are a treatment for leukemia as well. Because it is not an easy treatment, right? You irradiate your body so that your body doesn't produce any more blood cells. So your bone marrow is
So that you
irradiated.
can receive the new ones.
So you can
instead.
receive the new ones, and those can graft in, and your body can start making those. And this particular patient is really important because she identifies as mixed race, which is even more challenging for doctors because in the other cases of HIV cure in white people, they have been able to find a donor that matches both the person and then also has this mutation for the CCR5 protein that allows you to not get infected by HIV. So CCR5 is one of the major ligands that HIV binds to get into your cells in your body. So first of all, it's just harder for non-white people to find donor matches anyway. It's just the way that it kind of is right now. And then additionally, with this particular mutation, it's even harder. So this mutation is only found in 1% of white people, and it's even more rare in other populations. So the way that the doctors for this woman sort of got around this is they used umbilical cord stem cells and introduced the deletion, the mutation, and then also put in stem cells from family members that those cells would sort of mix together, and she wouldn't reject it wholesale, right? So if you don't know, umbilical cord stem cells are the most stemmy of the stem cells. They're completely programmable. They can become anything because they are essentially sort of blank slates.
Yeah.
Right. And it seems, so it's storming outside, but we're still sipping and sciencing in here.
Yeah, sure are.
So the engraftment took, which is the first big hurdle for this treatment, regardless if it's for blood cancer or for HIV. And this woman has been healthy and cancer-free and HIV-free since 2017.
Pretty amazing.
It is amazing, and it's hard, I think, because it shows a way that you could cure the disease, but it's a way that really isn't available to everybody.
Yeah.
It's only available to a select number of people, but hopefully what it does do is to give researchers and scientists clues on how they can eventually come about a cure for HIV.
With HIV, I saw one area that was consistent with with the research in stem cell, with the protein receptor, or I guess with the receptors, the DNA receptors for HIV.
No, not DNA, just receptors.
Just receptors. Okay. So
nothing to do with HIV doesn't have DNA. It's
Okay.
a retrovirus because it codes itself off of RNA.
yes.
Anyway.
See, Be advised. From one of the things that I was noticing in the paper was that part of the research that led to LDL receptor research was studying people who had genetically unable to create cholesterol.
Oh, that's an
And
interesting problem to have.
well, the work in attempting to understand genetically what was happening was part of the way that scientists eventually started thinking towards receptors.
I mean, it's interesting. So there's people that don't make it, and I guess the question is, do they have any impact? Like, what is the role of cholesterol in your body?
Well, cholesterol helps to build a lot of structures, including your vascular structure in your body. And that for people who have extremely low levels of cholesterol, their internal organs don't develop quite right and actually can suffer from a significant inability to thrive.
Yeah, I mean, it's funny. The cholesterol ends up being like most things in life, where it's really about moderation, and there's this happy range where things function optimally. And what's always been interesting to me is that we as humans are not hardwired towards that, right? We're hardwired more towards extremes necessarily. Like that lovely happy medium place is not something we do fairly well.
But as you get older, things don't work quite the way they did when you were younger, and part of that is your body's ability to manage how cholesterol is present in the blood, or even blood sugar.
And also, like we said in the beginning, there's a lot of genetic factors that determine your cholesterol metabolism, and so reasons why some people will get high cholesterol as they get older and some people will not, even completely irrespective of diet. All right. Well, hopefully we won't blow down or blow away with this storm. Cheers. And we hope you all have a great weekend. Cheers.